So our cladogram is an hypothesis, as all phylogenetic trees and cladograms are, but we base them on the evidence that we have, the morphological similarities between organisms, the molecular similarities between organisms. And I think that you did a really good job putting together a cladogram. So when we're looking at chronology, we're looking at when certain organisms arrived on the planet, or not when they arrived on the planet, but when they arose, and when and where they originated, but especially when they originated. That's all about chronology. And it's represented by the branching of the tree. So the farther back you go in the branching, the farther back that was chronologically. So what we've been looking at so far is just a relative chronology. So we figured that the tuna type animal would have been had an ancestor with later animals, but before the kangaroo and the other mammal branched off. So we're looking at relative rather than absolute chronology. What can we do about that? Well, we can use DNA. So this is a phylogram. And what the lengths reflect here are genetic changes that have taken place in very particular DNA or RNA sequence in that lineage. So the Drosophila is quite far removed, say, from the mouse, but a little bit closer to the lancelet. So these are the number of changes that have occurred, branching off. And the length gives you not a chronology in this case, but just a difference in DNA sequencing. So how different that might be. But an ultrametric tree gives you a pattern that's the same as the phylogram, but the branches can be traced to a common ancestor. And they're of equal length, because what we're looking at here is not the differences in DNA in a quantitative way, but we're looking at the differences that lead to a common ancestor sometime in the past. And we can verify some of these dates by looking at fossils. So fossils can verify branching points sometimes and give us an idea of the absolute time when this branching may have occurred. Yeah, so for example, here in the Paleozoic, about 500, no, not that long, about 400 million years ago, the amphibians branched off. So ultrametric trees can give you chronology, absolute chronology, but you need to have a lot of evidence. So building phylogenetic trees is about getting evidence. So if you want to get a little bit more complicated about it, we're going to get simpler. Parsimony means the simplest route, the simplest. And maximum likelihood means just that, the likeliest outcome. Maximum likelihood is all about statistics. Parsimony is making the simplest relationships. So you can never be sure of finding the single best tree in a large data set. Say you had 50 species and you were trying to arrange them in some kind of phylogenetic tree. Well, the number of possibilities that there are immense. I wrote them down somewhere. 3 times 10 to the 76. Those are the possible numbers of phylogenetic trees you could put together just based on ordering them randomly. So there are a lot of possibilities. So we try to find the simplest way of putting together our tree. So we'll put together, for example, the most similar looking organisms to be the most closely related. And organisms that don't look quite the same, that have some dissimilarities, we'll make them more distantly related. But the most parsimonious tree is the one that requires the fewest evolutionary events to have occurred in the form of shared derived characteristics. So where there haven't been changes, say, from limbs to fins and then back to limbs again. So we apply parsimony to a problem. It's fairly easy in molecular systematics. It's easier when you're looking at quantitative things rather than qualitative. So we can look at the similarities between humans and humans. Well, there are no differences. Humans are like other humans. That's all there is to it. How similar are mushrooms to humans? About 30% difference. Tulips, about 40% difference. So these are the percentage differences between sequences of DNA in very particular places. In the genomes. So we're trying to form a tree based on these 30% and 40% differences. Well, we're looking for the simplest way of putting it together in a phylogenetic tree. So it's more likely that the human and the fungi, since they're 30% different, will be closer than the flowers, because the flowers are 20% different. We're going to put those a little bit further out on the tree. And that's a simple kind of arrangement. This arrangement is less likely. It's more complicated. We're looking at the distance of the flower, and it doesn't make as much sense. So all we're trying to do is make the simplest relationships. And maximum likelihood, that's a tough one. It just means that given certain rules that we know about how DNA changes over time, whoopsie, a tree can be found that reflects the most likely sequence of evolutionary events. So you would expect that closely related individuals don't have too many differences between their DNA, but a more distantly related individual would have a lot of differences. So how do you apply that? I know this looks like a complicated thing. It is complicated, but maximum likelihood means that you get all of your phylogenetic trees that are possible and choose the one that's the most statistically likely to occur. So in this case, we're using statistics. Statistics to find the most likely tree. So it's important to remember that, see, it gets really complicated, that the best hypotheses for a phylogenetic tree, and this is like the crux of the whole thing, is the hypotheses that fit the most data. And we don't have all the data for everything, of course not, but you can look at different data and put them together. You can look at morphological data, what do organisms look like? You can look at their molecular data, how similar are their DNA? You can look at fossil data. Is there any evidence of common ancestors? So the phylogenetic tree, the one that is the best, will be the one that fits the most data. And it's interesting because sometimes there's compelling evidence that the best hypothesis is not the most parsimonious, or not the simplest. So it used to be thought that birds and mammals were more closely related than either to lizards, because birds and mammals both have a four-chambered heart. The mammal bird clade. But as it turns out, that's not the case. The lizard is more closely related to the bird. A different kind of clade, a lizard bird clade. It's not based on the four-chambered heart. It's based on evidence that comes from DNA and other morphological features that outweigh the four-chambered heart. And evolutionary history is documented in a genome. We can compare nucleic acids or other molecules like proteins, for example. It's a very valuable tool for tracing evolutionary history because you would expect closely related individuals to have very similar DNA. So for humans, we are, what is it, 0.01% different from each other. So 1 in 3 million base pairs will differ between you and I. But between humans and chimpanzees, it's 0.1%. And so we know that because you can extract DNA and you can sequence it and you can see exactly what the sequences are. So it's a very valuable tool and it's used quite a lot. And so the rest of the lecture goes into molecular clocks and sort of convoluted stuff that I don't really think we need to lecture about. I think we have a good handle on how to build a phylogenetic tree. So that's basically what this was all about. How to build a phylogenetic tree. Gather evidence in the form of morphological similarities, molecular similarities, and differences, of course. Fossil evidence, and make the simplest relationships based on your evidence. And remember that phylogenetic trees are hypotheses. So they're pretty good. Often they're pretty good hypotheses, but sometimes they're overturned, like the whole birds and mammals being more closely related because of the four-chambered heart. So sometimes those kinds of things are overturned when more evidence becomes available. All right, that's really all I wanted to say. Thank you very much for watching.